The diencephalon can be included with the anterior part of the brainstem. It is formed from the prosencephalon, the hypothalamus and subthalamus being older portions. The thalamus, metathalamus, epithalamus are the newer parts. They are referred to as thalamencephalon. The cavity of the diencephalon is the third ventricle. In this sagittal section, the diencephalon is indicated in blue. In this image, we see the early stages of brain development in the embryonic period. At the stage of 3 cerebral vesicles, 25 days after conception, diencephalon doesn't exist as a separate brain structure. It appears after 3 days as a result of differentiation of the forebrain or prosencephalon into ultimate or final brain or telencephalon and the intermediate brain or diencephalon. Here it is. In this diagram, we see stages of 3 and 5 cerebral vesicles and their derivatives. So, derivative of the cavity of the diencephalon is third ventricle. And the definitive brain structures of diencephalon are the thalamus, epiphysis, corpus geniculatum laterale, corpus geniculatum mediale, hypothalamus, which includes corpora mammillaria, tuber cinereum, infundibulum, chiasma opticum, and tractus opticus. Now, classification of the structures of the diencephalon. Diencephalon consists of thalamencephalon and hypothalamus. Thalamencephalon includes thalamus, subcortical center of all kinds of sensitivity, epithalamus, and metathalamus. Now we see complicated horizontal section of the brain, dorsal view. So why the section is complicated? Because to prepare such a specimen, you need to make 2 horizontal sections. First of them is lower layer. It passes through the temporal lobe and occipital lobe. Temporal lobe and occipital lobe. Due to this section, we can see both lateral ventricles, the left one and right one. Second section is made more superiorly through the corpus callosum. Here it is, and here it is. Due to this section, we can see cavity of third ventricle. Here it is. Now, the red rectangle demonstrates us the diencephalon and the adjacent structures. So we need to distinguish and remember all anatomical structures in the region. Ready? Now I use a part of previous picture to analyze all anatomical structures. So, what is number 1? It's corpus callosum. Through it passes the upper layer of this specimen. What is number 2? Cavum septi pellucidi. So it's narrow cavity between two septum pellucidum, right and left. Number 3 is cornuanterius ventriculolateralis. Number 4, caput nuclei caudati. Number 5 is columna fornicis, structure which belongs to telencephalon. Number 6, adhesio interthalamica, the structure which connects to thalamus between each other. Number 7, corpus nuclei caudati. Number 8, ventriculus tertius. Number 9, stria medullaris thalami, narrow white matter line which goes on the periphery of the thalamus. Number 10, lamina fixa and stria terminalis, as prolongation of stria medullaris. Then number 11, cauda nuclei caudati. Number 12, pulvinar thalami, posterior part of the thalamus. Number 13, corpus pineali or epiphysis. Number 14, colliculus inferior mesencephaly. Number 15, colliculus superior mesencephaly. Number 16 is trigonum habenule. And number 17 is habenula. Remember that habenula translates into English like leash, for dog leash, for example. And number 18, commissura habenularum, which connects habenula from left and right sides. Number 19, tuberculum anterius thalami. And at least number 20, commissura fornicis. Now that we know the basic anatomical structures of the thalamencephalon, let's compare this picture from the anatomical atlas with the schematic diagram of the same structures. The blue structure in the center of diagram represents the thalamus and corresponds to the designation number 19, tuberculum anterior, and 12, pulvinar thalami, from the anatomical atlas of Sinelnikov. At the periphery, the thalamus is surrounded by strips of white matter, SM, stria medullaris medially. It corresponds to number 9 in the photo. Here it is, stria medullaris. And ST, number 10, stria terminalis laterally, which corresponds to number 9 in the photo. stria terminalis separates the thalamus from the nucleus caudatus, and C, it corresponds to number 4, 7, and 11 in the photo. Nucleus caudatus is here, and in our scheme is here. Near the median plane, after fusion stria medullaris and stria terminalis with each other, a flat triangular area called the triangle of the leash, or trigonum habenula in Latin, designated as TH in our scheme, and number 16 in the photo, is formed. Thin leash, or habenula in Latin, departs from this triangle and fixes the pineal body or epiphysis, designated as E in our scheme, or number 13 in the photo, from both sides. Here they are, habenula and habenula in the photo. The epiphysis thus finds itself between the two upper collicles of the four-collical plate of the roof of the midbrain, colliculus superior, CS, or number 15 in the photo. In this regard, epiphysis, sometimes called as fifth collicle of the four-collical plate. Here it is, between superior collicles, and it looks like fifth collicle in four-collical plate. Now that we know the basic structures of thalamic part of the brain, let's draw together a schematic diagram of thalamencephalon. So, TH is a thalamus, right and left. A is adhesio interthalamica, the structure which connects two thalamus between each other. V3 is ventriculus tertius, or third ventricle. Pulvinar thalami is the posterior part of the thalamus. And now we divide left thalamus on the groups of the nuclei. A are the nuclei anterioris thalami, which are subcortical centers of the smell. B are the nuclei posteriores thalami, which are the subcortical centers of vision. N, M, L, nuclei ventrolaterales, which are subcortical centers of the general sensitivity. NM, nuclei mediales, which are subcortical centers of extrapyramidal system. and at least an I nuclei interstitiales which have vestibular and auditory function. Now NC nucleus caudatus, which is situated laterally to the thalamus. It belongs to the Diencephalon. And now thalamus is surrounded by these yellow strips. SM stria medullaris and ST stria terminalis, which divides the thalamus and nucleus caudatus. And now epiphysis, which is hanging on the H habenula and on the EH trigonum habenula. So we can say that these three structures, epiphysis or corpus pineale, habenula, and trigonum habenula, belongs to the epithalamus. And now another structures. CGM, corpus giniculatum mediale, and CGL, corpus giniculatum laterale, which are connected with BCS, brachium caliculi superioris, and BCI, brachium caliculi inferioris, with lamina quadrigemina. So they together belongs to the metathalamus. Now let us find all these anatomical structures in another picture. This is dorsal surface of the brainstem, and I'm using this black rectangle to zoom in. So letter E means epiphysis. This arrow H, habenula, or leash. The H is trigonum habenula. All these three structures belongs to epithalamus. Now CGM is corpus giniculatum mediale, which is subcortical auditory center. And CGL is corpus giniculatum laterale, which is subcortical visual center. And BCI is brachium caliculi inferioris, and BCS in parenthesis, brachium caliculi superioris. Why in parenthesis? Because it's absent in this image. So I use another picture to find it. BCS is here. It connects caliculi superior and CGL, corpus giniculatum laterale. And here BCI, brachium caliculi inferioris, which connects caliculus inferior with CGM. corpus giniculatum mediale. And now all these four structures, two geniculate bodies and two brachia, they together forms metathalamus. Third ventricle. Third ventricle is the cavity of the Diencephalon. So third ventricle is very narrow cavity between two thalami, right and left. In this picture, we can see that third ventricle is connected with fourth ventricle through the aqueductus cerebri and with left and right lateral ventricles through the foramina interventricularia or foramen of Monro. This is a sagittal section of the brainstem, and we use it for demonstrate the red rectangle, the middle part of the diencephalon, and now we shall zoom in the structure. So it's the same structures. And now we shall find all the walls of third ventricle. First of all, anterior wall, which is formed by lamina terminalis. Here it is, very narrow structure, which is derived from the wall of prosencephalon, first cerebral vesicle. Now, inferior wall of blue color consists of chiasma opticum, number 1, tuber cinereum, number 2, corpora mammillaria, number 3, and substantia perforata posterior, number 4. To better understand all the structures, let us also use the inferior aspect of the brainstem. So here they are: chiasma opticum, CHO, tuber cinereum, TC, corpora mammillaria, CM, and substantia perforata posterior, SPP. Now, superior wall, which is roof of third ventricle. It is represented by telechoreida ventriculi terzi. Here it is, telechoreida ventriculi terzi. We remember that it is connected with telechoreida ventriculi lateralis through this opening, foramen of Monro. And that's why telechoreida ventriculi terzi and telechoreida ventriculi lateralis are practically the same structure, but in third ventricle it make a roof or superior wall of the ventricle. And now the posterior wall, this yellow, it consists of two structures, commissura posterior, here it is, and number 2, commissura habinularum. So all of them make walls of third ventricle. Now the hypothalamus. Here it is sagittal section of the brainstem. I use this red rectangle to zoom in the image. So now we can classify the hypothalamus. The hypothalamus is classified as follows. There are 4 regions of hypothalamus. A. Regio hypothalamic anterior, so pars optica. It consists of number 1, chiasma opticum, and number 2, tractus opticus. The next region is regio hypothalamica intermedia, B. It consists of regia subthalamica propria, RSP, and number 2, tuber cinereum, and number 3, infundibulum. Attention that infundibulum and tuber cinereum practically are the same structure, viewed from the inside, from the cavity of the third ventricle, and from the outside, from the basis of the brain. So practically, they are the same structure. And now number 4, hypophysis or pituitary gland. Please note that the pituitary gland is depicted in 2 colors. The posterior lobe of the pituitary gland, light in color, is a continuation of the infundibulum and is connected to it by means of stalk. This is narrow hypophysis. It develops in embryonic period from the Diencephalon. The anterior lobe is depicted in dark brown color. This is adenohypophysis. The anterior lobe, or adenohypophysis, develops from the epithelium of the dorsal wall of the oral cavity in the form of finger-shaped outgrowth, so-called Rathke pocket. This ectodermal protrusion grows towards the bottom of the future third ventricle. Thus, the 2 lobes of the pituitary gland have different embryonic origin, and as we will see later, their hormonal production is organized differently. Next classification group, regio hypothalamica posterior. So, pars mammillaris of hypothalamy, or we can say corpora mammillaria. And last region of hypothalamus is regio hypothalamica dorsolateralis, which is presented by nucleus hypothalamicus posterior or nucleus Lewisi. Now let us find the same structures on the basis of the brain. So, corpora mammillaria, CM, then we depict PC, tuber cinereum, and infundibulum. Attention on the stalk of the pituitary gland. Then we describe PO, tractus opticus, and CHO, chiasma opticum. And the most posterior part is the substantia perforata posterior. Here it is. And now nuclei of hypothalamus. Let us make together the consecutive drawing of this picture. So number 1 is commissura anterior. Number 2 is chiasma opticum. Number 15 is lamina terminalis. Number 3, hypophysis or pituitary gland. Number 4, corpus mammillare. And number 5 is sulcus hypothalamicus, which is a border between thalamus and hypothalamus. Number 6 is infundibulum. And number 7 is recessus infundibuli of third ventricle. And now nuclei. Here they are. Number 8, nuclei infundibulares. And number 9, nuclei viralis. Now, number 10, nucleus hypothalamicus anterior or ventromedialis. Number 11, nucleus supraopticus. Number 12, nucleus paraventricularis. Number 13, nucleus hypothalamicus dorsomedialis. And number 14, regio or area hypothalamica dorsalis. Now we combine the two nuclei, nucleus supraopticus and nucleus paraventricularis, into a group of large cell nuclei, so-called magnocellular system. The other two nuclei, nucleus hypothalamicus ventromedialis and nucleus hypothalamicus dorsomedialis, are combined into a group of small cells nuclei, so-called parvicellular system. Now we will look at the hypothalamus-hypophysis connections and how hormones are produced from these groups of nuclei. So, magnocellular system. Target is posterior lobe of hypophysis via supraoptica hypophyseal tract. That means nuclei paraventricularis and supraopticus. Pituitary hormones are not synthesized in the posterior pituitary lobe, but in neurons located in the paraventricular nucleus and supraoptic nucleus of hypothalamus. They are transported by axons of the hypothalamic hypophyseal tract to the nearer hypophysis, where they are released as needed. Terminals of paraventricular and supraoptic nuclei release two types of hormones in the posterior pituitary lobe: oxytocin from the paraventricular nucleus and antidiuretic hormone or vasopressin from the neurons of supraoptic nucleus. The axons from both nuclei pass through pituitary stalk to the posterior lobe of the pituitary gland. The peptide hormones are accumulated in special vesicles aggregated into herring bodies in the axons of neurosecretory neurons and are carried to the posterior hypophyseal lobe by means of axoplasmic transport. And now parvicellular system. Target is portal system of median eminence via tuberar hypophyseal tract. The superior hypophyseal artery from each side of the body form a vascular plexus around the infundibulum or pituitary stalk. The axons from neurons of the hypothalamic nuclei terminate at the plexus and secrete hormones that have been produced in smaller parvocellular neurons of the hypothalamus. The secreted hypothalamic hormones are of two types: releasing factors which stimulate hormone release from cells of the anterior pituitary lobe, and release inhibiting factors which inhibit release from these cells. These hormones are carried by hypophyseal portal venous system, named after the portal circulation of the liver, to capillaries in the anterior lobe, establishing communication between the hypothalamus and endocrine cells of the anterior pituitary. And now functions of hypothalamus. The most important functions: control of homeostasis. It includes thermoregulation, osmoregulation, and glucoregulation. Very important function: circadian rhythm, sleep and wake cycle. Next function is stress reaction, hypothalamic hypophyseal-adrenal axis. Number 4, autonomic nervous system. Here is the main autonomic nervous system control center. Control of emotions and control of reproduction.